Thank you for this opportunity to talk to you today. Let me just tell you a little bit about my work, and let me introduce you so you know a little bit about me. My grandfather was twice elected the national president of the equivalent of the county agents, basically with ag extension. My other grandfather was in cattle. We ran about 1,000 head of cattle. The area we ran it on was so large that he branded me a calf when I was 10, and we found it when I was 18 and going to college. So we sold it, and that paid for tuition the first year. Now, I grew up in a little town called Green River, Utah. I had 13 in my high school graduating class, and the area there, we call that 100-mile-an-hour range because that's about how fast the cows have got to be going to get enough to eat. That's not actually my birthplace, but anyway, it gives you an idea of what it looked like. I did my bachelor's and master's in botany and range science. So I'm basically a plant ecologist. I've worked at the community level and the ecosystem level and done a lot of work on invasive species in the past. But working for the Bureau of Land Management out sampling rangelands was a little bit like trying to drain the ocean with a teaspoon. You'd throw the hoops down and you knew that that was not really representing the 1,000,000 acres you were going to apply it to. So I decided that I needed to find a better way to look at plants and inventory plants, and that's how I got into remote sensing. Now, I started at Brigham Young University. I did my PhD in geography, in biogeography, at the University of Utah. Now, I show you these slides for a reason. You see the three-dimensional component of the environment I was working in. There are mountains there. I was in the Rockies. Moving to Kansas, I found a very different situation, obviously. Now, one of the things about remote sensing is that topography plays a very, very critical role in the spectral reflectance of the target you're looking at. Here you're looking at sand dunes in White Sands, New Mexico. And I'm betting that none of you would argue that the sand right here is different than the sand right there. But it is spectrally different because of topography. We call this the topographic effect. And for years, working in the Rockies, I tried to figure out how to get rid of the topographic effect. I can see Christina knows what's coming next. So I finally found a solution after pulling my hair out for many years, and that is I moved to Kansas, all right, so that I got rid of the topographic effect. All right, but then what I found out is there's microtopography. But I'm learning how to use that microtopography to characterize plants based upon their canopy geometry, the structure, the way they're orienting their leaves, and so on. So we'll show you some of that in a bit. Now, I went to Kansas, University of Kansas in Lawrence, and taught there for 19 years. I was over the Kansas Applied Remote Sensing program. And then I moved to Kansas State. I wanted to get back to my ag roots, and so I taught in agronomy for 5 years. But while I was in agronomy, I was trying to show the agronomist satellite imagery, how cool it was. And they didn't yawn on my face, but it was close to that sometimes, you know. It just didn't turn them on. Finally, I decided, well, maybe I need to make the pixels smaller, these picture elements smaller. So what I did is that I put a camera on a plane. I spent $100,000 figuring out what works and what doesn't work in terms of these drones. We call them drones. Some people say you're supposed to call them small unmanned aircraft systems. Most of you in the room would know I was talking about drones. I was just trying to tease or fool you, right? So the difference is that a drone is autonomous. It can be flown. You put it in the air and it'll just fly for you. So at Kansas State, I started flying drones and then showing the agronomist imagery now, instead of being 1 kilometer by 1 kilometer or 30 meters or 20 meters by 20 meters or whatever, now I was talking about 3 millimeters pixels. I was looking at individual leaves and looking at the reflectance characteristics. So we'll show you some of that in a bit too. Okay, I'm going to teach you a little bit about drones. I'm not here to sell you a drone, so relax, okay? I'm not trying to sell you a drone, but I am going to show you ours because that's the way that's the way I'm going to show you some of the things that I think are important to have on a drone if you're going to get into this. And I would advise you not to get into it until you've done a lot of homework, because there's a lot of ways to lose a lot of money in this field. All right? But basically, the drones fall into two categories: what we call fixed wing, which is like the plane, or multi-rotor, which is multiple blades that fly. The multi-rotors are very easy to fly. Everyone in the room could be flying one in 5 minutes. You may lose it if you don't know what you're doing, but you could be flying it, all right? The fixed wing, they're like an airplane. I found from experiment that a delta wing like this, one that has a low profile, doesn't have the high tail fin and all that, handles a lot better in the wind. We have flown this particular aircraft with a tailwind at over 100 miles an hour, and the wings didn't fly off, all right? So that was a good sign. Let me show you a little more about them. I'm going to take you into showing you the multi-rotors are really great for doing research over small areas, like a few hectares. You can cover 50 acres, convert that to hectares. I can't remember what that number is, but we're still in acres there in the U.S. But anyway, you can fly about 50 acres in a day with the multi-rotor. They can fly for about 12 minutes to about 25 minutes, depending on the aircraft. You can take them and fly them very low and very slow, so you can get lots of good detailed data for study plots and so on. So if I were doing research on smaller plots, I would use a multi-rotor. And I'm going to show you a video of us doing some high-throughput phenotyping on winter wheat using a multi-rotor. Here's a couple of my graduate students. And I'll kind of talk while it's going. So you notice that first off, you notice green and pink or orange styrofoam on the bottom. That's so when you're flying it, if you accidentally turn it around while it's flying, you know which way it's oriented, and you can turn it back around so you don't fly it away from you instead of toward you when you these are winter wheat genetic study plots. They were supposed to be all lined up real nicely, but the GPS didn't work that day, I don't think. I didn't do that. Now we're flying a color infrared camera. You can send a regular camera in and have it converted to capture color infrared imagery. So for about $1,000 to $7,000, depending on the camera quality. So there's a color infrared with some calibration plots showing you that winter wheat. We got down low enough that we could count seed heads if you wanted to count seed heads, and we weren't counting kernels, but we were counting seed heads. All right, that's probably enough of that one. This one is a fixed wing. With this one, we can fly about 1,000 acres a day. And this aircraft is made out of car bumper foam. So we've crashed this from 200 feet up, picked it up, put it back in the air. This one, we built this one, so it's not a hobby plane that's been converted. We actually designed this. It cost us $400,000 for this. To give you an idea how expensive this stuff can be, that is made out of EPP 2.2 density foam. Notice it's got multiple bays, so you can put multiple sensors in it. It's 70 inches wide, so it flies flat and much more level. It's not doing this all over the place. So you can get better imagery with it. This was built for our dealers, so we're not out trying to sell this plane to others. We just wanted to have an aircraft that our dealers could work with. Now you're going to see it launched. It has a big bungee cord that's hooked to it, and that saves a lot of money. If you have a catapult, they're about $2,500. Our bungee cord is $39.95. All right. So the fins on it, they're made so that there's skid plates underneath. So when you land it, it skids on the plates. And when you wear the plates out, you go to the hobby store and you can buy some more and replace them for a couple of bucks. So the wings are made so you can take them off and replace them if you break them. Some people will tell you that you need to coat your wings with fiberglass or carbon fiber or Kevlar. Let me tell you something. The heavier it is, the worse it flies. When you're bringing it into land, you have a stall speed when it's heavier, and it stalls more quickly, and so it just drops out of the air on you. When you crash these things from 200 feet up, I don't care what it's got on it, it's going to blow up, all right? So you just don't. The better thing is to have a light aircraft. Don't gobbledygook it all up with all this stuff, because Kevlar, all that just breaks and shatters when you hit the ground that speed. No landing gear on them. The reason there's no landing gear is because if you put landing gear on them, they do somersaults when you land them, because they get hooked in the grass and the weeds. So you just belly skid them like this and pick them up and take your camera out. All right, Christina works here in this area. She's one of our dealers, and she flies the RF-70. That particular overflight you see right here is 1,424 pictures were taken. She flew for about an hour and covered, I can't remember how many acres. You remember how many that is about? How many? About 320? Okay. 320 acres in about an hour. The resolution on this is probably close to 2 centimeters, is my guess, somewhere in that range. All right, so notice the flight lines and so on. But, okay, you're not going to hear this from any other people who work in drones. I'm going to tell you that drones are a lot more expensive than flying a Cessna. And that freaks people out because they say, how can that be? And because a lot of people charge more for the drones than for the Cessna. And the other reason is because you've got to have 2 people flying it. To be legal in the U.S., you have to have a spotter. You can't fly it more than a half a mile away because it goes out of sight. You have to keep it in line of sight. If you're commercial, it can't fly higher than 200 feet. You can fly 400 feet in some instances, but usually 200 feet. So you're going to fly a lot of flight lines. And the batteries run down on you. The batteries are not cheap. You're talking a couple of $100 per battery. It's worse than the car battery for buying them now and so on. So it's not the answer to cheaper. It's just a different tool that's used for different purposes, okay? But with the Cessna, we can fly 20 to 30,000 acres, and with the new cameras that are coming out that are 50 megapixels, we can fly at about 10,000 feet and still get about 10-inch resolution, and can fly over 50,000 acres in 45 minutes. So I just want to be very, very upfront with you that drones are not always the best solution. And in fact, when the fun wears off, it becomes work, all right? Just so you know. All right, let's talk about remote sensing real quick, and then I'll show you some examples. This is the way a dandelion looks to us, the yellow one. The other one is the way it looks to a honeybee. They're seeing things in ultraviolet. There are wavelengths that we cannot detect with our eyes that are still very valuable. See, that tells the honeybee right where the nectar is, basically zero in. So remote sensing is sensing something without being in physical contact with it. And your eye is one of the best remote sensing tools there is. But when we're looking at, in the field that we work in with satellite imagery or with drone imagery, really we have an index I'm going to introduce you to in just a second called the Normalized Difference Vegetation Index. There's lots of indices, but this is the one that's used the most. All it's doing is detecting differences in chlorophyll density. So those chloroplasts are like little antennas that are sucking up wavelengths in the blue and in the red for photosynthesis, and they're reflecting away a lot of the near infrared. So here's a soybean plant in the greenhouse. Part of it is sick. The side's sick. Right there, it's still a little bit healthy. Dark green. I'm going to let you look at it. There it is. If I handed you a pair of glasses that could only detect blue wavelengths, green wavelengths, red wavelengths, or near-infrared wavelengths, that's the way that plant would look. And notice that when it's healthy, let me go back. Where it's healthy, it's dark in the visible because it's absorbing that light for photosynthesis. And where it's in the near-infrared, it's bright. It's reflecting a lot of that light. It's a little hard to see, but there's dead leaves on the table. The dead leaves will look a lot like the live leaves in the near-infrared. But in the red band, the dead leaves on the table are bright, and the healthy leaves are dark. Let's see how we use that. We simply take the near-infrared and subtract from it the red. Now, these pictures are made up of millions of little numbers, and so we're numerically subtracting red from near infrared, and then we're normalizing it by taking the product of the red and the near infrared and dividing it into the difference, and you get what's called the NDVI, and that's what this is. Now notice that the healthy part of the plant is bright, and the sick part of the plant is dark, and that's because it is the healthy part has a high NDVI value, and the unhealthy part has a low NDVI value. We colorize it, so here you can see we've said, okay, anything that's healthier, make it green, anything that's sick, make it red, and now we've got a map. We can fly the entire field for every 2 centimeters square in the field, or 4 centimeters square. For every 4 centimeters square, 2 centimeter by 2 centimeter area, you've got an index telling you, giving you an NDVI value. Okay, I'm going to talk to you about some of the earlier work I did in satellite remote sensing on weeds, invasive species, and I hope the one I'm going to show you is legitimate. You may not have it around here, but I'm sorry I didn't have any. One of them that's a big one in invasion is the juniper trees. And there's multiple species of juniper. This one is called Juniperus osteosperma, Utah juniper. I did my PhD work on this one. If you want to get rid of it, the best way to get rid of it is throw a match on it, but you better have someone's permission, because there's no such thing as a controlled burn with juniper, all right? It's oh my gosh. What have I done, all right? So you don't throw a match on this unless you've got it all set up right and got a lot of people there with fire engines and all that. But the juniper takes a lot of the moisture out of the ground and kills out all this understory. So we lose a lot of plant life. Here's some before and after, 1905 versus 2000 on the right. And some of my work was looking at how the different densities of trees influence soil erosion. So what I've got there is basically a graph showing that the soil erosion starts very early in the invasion. People are going out studying it after you had full canopies, and they weren't finding that much erosion. The reason they weren't finding is because all the erosion had already taken place. The ground was covered with rock material and all that was armoring the top, and so erosion slowed down. You can see by the time it gets over to these later stages of the development, the initial rate of erosion and the end erode are about the same. You have to catch it when it's first starting to be invaded if you want to know how the effect of erosion. We were able to use satellite data to predict the percent cover by trees, so that graph showing it. There's something here. It's just the opposite of what you're taught. If you ever took a remote sensing class, you were told that with increased plant material, you get an increase in near infrared. It's just the opposite, because with the trees, we're getting an increase in shadow. So the shadow component is playing a big part in affecting this relationship until it hits 50% cover. At 50% cover, the relationship starts to turn around, but I didn't have any site that had more than about 60% cover, so it didn't go any higher. When I got to Utah State, one of the first projects I worked on was mapping Dyer's Road, a mustard plant that was invading. The students went out and inventoried every spot they could find this in multiple canyons. They rode horses and they walked to find this. And so the first map is showing, you can't see it, little red dots. We then took those red dots and we used those to train the computer to show us all the areas in those mountain ranges that were susceptible to invasion by Dyer's Road. And the yellow areas are the areas that have a higher propensity to be invaded by this plant. That map got some people off top dead center and started throwing money at it for controlling it, because they realized that much of that whole area was susceptible to invasion. This happens to be a wilderness area showing that it could basically be wiped clean by just this particular plant. In Kansas, in the central Great Plains, we have another juniper called eastern red cedar, and this one is taking over and wiping out the grasslands, the native grasslands and so on. So we're trying to control it also. The trick is knowing how much you've got. This little map is just showing some work that we did with the Forest Service. The red areas are where the red cedar was located. So we were able to map it from satellite quite easily, but I want to show you how fast it's invading. This is Douglas County in Kansas. There's a lake right here, and it's between 1985 and 2002. This is what it looked like in 1985. The red area is the juniper. There's 1990, 1995, 2000, 2002. They say a picture's worth 1,000 words. That got people interested in invasion. If you can show them what's happening, all at once they begin to realize there's a problem. It's not just a bunch of scientists in there trying to them, you're just here trying to get more money, right? You got to prove to them that something's happening. And a map, a photograph is very powerful in doing that. Okay, now let's go to the ultra-high resolution imagery. We did this at 1 meter resolution. We developed what we call a weediness index in one of the study areas, and we looked at the relationship between the spectral data and found a fairly decent relationship, R squared 0.78, on predicting the weediness. Now, you're saying, how did they detect, or how did you classify it? We used the weediness of the plants was based upon a floristic quality assessment and an index. It was called the coefficient of conservatism. And basically, it's an index that tells you, that classifies the plants according to how aggressive they are for invasion. And so we used that, and that's how we built all the plants in our study plots. We classified them into categories. Thank you for sending us Canada thistle. We appreciate that a lot. Of course, we probably sent you some of our plants too, right? All right. Well, anyway, this farmer was told by his agronomist that he had Canada thistle in the field. In the U.S., it's against the law to let Canada thistle grow in the field, unless, of course, they've got a passport or something like that. But I'm just kidding. But what the farmer did then is he hooked up his sprayer, and he went out and sprayed 120 acres on this pivot to kill Canada thistle. Do you notice any interesting thing about it? This is drone imagery, and there's a clump of Canada thistle, there's a clump there. See, it's a cloner. So it's not spreading everywhere, it's growing in clones. It's got rhizomes under the ground. And so the farmer didn't need to spray 120 acres, he only needed to spray 0.6 acres. So he did something that the environmentalists would not be happy about. He put a lot more chemical down than he needed to. But he also spent nearly $4,000 on spray when the imagery and the spray would have cost him about $500. So he spent a lot more money than he needed to. Now the story gets worse because a few days later we had 50-mile-an-hour winds, and his corn was up high enough that the spray he put on it caused the stalks to become brittle, and he lost 55 acres to green snap. So the green, well, that says 55. So basically it snaps them off and you're through. And he lost 55 acres. So that loss, when you put all of it together, the loss is about $41,000 on that field. Now let's compare the drone imagery and the Cessna imagery. I don't have a good comparison on Cessna on this one, but we could have flown this at just about the same resolution, a couple inches coarser with a Cessna. But here is that field, and this is the 1.5-inch resolution, and you see the weeds in here. If we use 1 meter satellite imagery, that's what that same area looks like. And if we use the 5 meter, like rapid eye satellite imagery, that's what it looks like. If I go to the next one and show you the NDVI, you can see the weeds very clearly. But where are the weeds and where are the weeds at that resolution? So if the farmer were using 1 meter satellite imagery, or you as an agronomist were, or if they were using the 5 meter, let alone 30 meter, the farmer still would have lost $41,000 because you needed to know where the weeds were in order to do a precision ag application. Here's from 130 meters up. You start looking, you can see it's a little hard in this lighting condition, but the weeds are right in here. There's some erosion, soil erosion. This is winter wheat. Here, any idea what the farmer did wrong here? Double planted. In other words, he planted winter wheat on top of winter wheat. In some areas that might be good. In this area, he planted weeds. And what weed was it? It was the same species of the weed that he was growing. What herbicide is he going to use to take that one out? So he basically reduced his production there because of it. Here's showing you some zoom in on it, where they're crossing in two different directions. And it was over dozens and dozens of acres that he did this. I took out the slides showing that just to save time, but that's at 130 meters. If you got a good camera on your plane, you can carry a good one. That's the level of detail. You can see how effective they are in getting the seeds planted. You can see some what we call litter or crop residue. You could start picking out weeds. The weeds don't usually grow right down the row with the crop. They kind of cheat. They grow in between sometimes. All right, so here is that field, a field next to the one I've been showing you, and you see the pattern there. The weeds are kind of splattered everywhere. Here we're seeing two types of growth forms, clustered and random. Random, what would that, you're the weed scientist. If you find the weeds randomly scattered in the field, what does that tell you about how it's probably being disseminated? Come on. Wind is my guess, okay? It's a wind-disseminated plant. It's probably an annual. If you got a cluster like that, it's not wind-disseminated, and it's probably a perennial. You use two different types of chemicals on it, depending upon the growth form, the life form you're dealing with. Here's a map showing the weeds in this field. The red is where the weeds are. We flew this field in 15 minutes. And we developed classifiers that go in and classify it. And we're working with some of the equipment dealerships. There's going to be a move to start building equipment that can do more precision ag and spray this stuff. Now they just spray the whole field. But if they had this kind of information, they wouldn't spray the whole field. We can put it into densities, different levels of density. So you tell the sprayers for this area, spray at this level, this area, spray at a different level, or you decide which of those squares you want to completely cover and you spray them. Yes?
So Kevin, is that a germination? So the whole area would be contaminated, but only those areas that germinated when the flight was taken?
This, I don't remember exactly how big the weeds were, but as I recall, they were a couple of inches tall. They weren't really tall, but and so the question is, how soon can you detect them? And we're working on trying to test that. We've got our dealers sending us in imagery at different stages. And by the way, the person who asked us to fly this went out and walked the whole field and said it was spot on in terms of classification. The weeds usually have a different growth form than the plant you're working with. Not always, but sometimes. So this is weeds again in another field. Do you notice anything about that one? Why do the weeds grow in lines? Compaction. Yeah. It's where they're running over it, and the tires are pressing the ground down around the weeds, and they like that. You know, they hold moisture better. So you can see how they're growing in really interesting lines. Now, one way we can discriminate between certain species types is using what I call a multi-temporal analysis approach. And that's where we analyze multiple overflights. From a research standpoint, this is something I have done in the past, but haven't done much lately. But I'm going to throw out the idea and let you think about it. What we're looking at here is an area that's forested area, grasslands, and grasslands. and there's a road to give you an idea of the scale. This is about 1 meter resolution imagery, flown when I was a professor. The red area is eastern red cedar. It's evergreen, so it's photosynthetically active, even in May or March. You see the trees have started to green up by May, and then in July, you see how they're spectrally changing. That's because of different phenologies. The plants have different phenological development stages. And so we can use this to tell one plant from another type. And you can see that this is the cool season grass. See, that's October. The only grasses that are photosynthetically active in October in our area are the cool season. Bromus inermis is out there growing like crazy. This one right here, that's warm season. So we've got two different, you've got C4 and C3 plants going. So you can start classifying them according to the way they fix carbon. And we've done that, and we found that if we do the wrong time if we fly our imagery at the wrong time, like about right there, this is the NDVI plotted over time over the growing season, and this is the top of a building that doesn't change, so we're showing that the variation we're seeing here is because of the plants, not reflectance. And you see right there, they're all crossing over. That's telling us that that's the wrong time to be capturing imagery. Everything is spectrally similar at that time. So you look for times when they're all different from one another to try to classify them. And it changes over the growing season, depending on what the plants are and what their mechanism is for fixing carbon and so on. Now, I had a graduate student. We only had money. Back when we did this, a satellite image cost $4,400. And we didn't have money to buy one at that time. So he used one that we had, and he was testing to see if a government program called the Conservation Reserve Program, if he could detect Conservation Reserve Program lands from lands that were tall grass and short grass and cool season, warm season. And the only image he had was in September. What do you think his conclusions were? He concluded that satellite imagery couldn't be used for discriminating between them. If it is used here or over here or a lot of other times, he would have got entirely different conclusions. This idea of being able to track the way the plants green up, this is the onset date of green. This is the NDVI plotted over a growing season. So this is the onset date, the rate of green up, the rate of brown down, how long it stayed green. We use those metrics, we call them vegetation phenology metrics, to characterize plants and tell one plant from another. We use it also to forecast yields on crops. We can forecast months ahead of the U.S. Department of Agriculture, and we're much more accurate by using an approach where we're monitoring. We monitor most of the world on a weekly basis to see how it's doing. You can do this same thing at a much smaller scale and start discriminating different plant types, whether they're weeds or not. This is showing corn, and the idea that's an actual curve, and we look to see whether or not that corn is greening up, how it's greening up. We found out when it hit that point, it's tasseling, it's going into reproductive mode. You can do the same thing. What stage of development is the plant at? You can look at texture. This is canola versus winter wheat. And if I put a transect through it, that line there is a transect. With our software, we pull out all the values and plot the variation in the reflectance characteristics of winter wheat versus canola and their textural differences. Here's some plots of soybean, and I pull up some data from three of those plots, put it into graphs, and you can start. Now, the way that I'm showing you here is that the data are numerical. It's not just a picture. You can pull numbers off it, put it into models, and use it for studying. Last one, last couple of slides here. I want to show you how we've used it to do high throughput phenotyping. And what we're doing is measuring the canopy geometry at the 3 millimeter level. So on sorghum, the sorghum is being bred to increase biomass for biofuel. And we wanted to know in those plots, the geneticists in the area we're working in have 33,000 plots. And they have to try to walk around and look at each plot to figure out what the phenotypes are. We're developing methods that will automate a lot of that process. So what you see is the level of detail. We're looking right down into the sorghum, and we're breaking it into classes. Lower level leaves, middle level leaves, upper level leaves, and the colors over here telling us how much area is at the different canopy levels, which is telling us something about the amount of light that's penetrating and how those plants can be using that for photosynthesis. We took all of those plots and analyzed them, and there's the graphics. Now, what does the graphics mean? The next slide will be better. I'm just showing you that it's numerical. What we're breeding for in this particular case is increased photosynthesis. And the way you increase it in this particular plant is get the upper leaves to grow more erect and the lower leaves more prostrate. That way they're letting more light down in deeper into the canopy. So we characterized them. Notice that by doing this, the colors that we're looking at over here, this particular tract, that plot had more of a vertical geometry, is because we're seeing more area down deep in the canopy. Over here, this is more erect. We'd select against these two, we'd select for those two. And that's it. Okay. Did I get done in time? All right.